So, hello everybody. We are very happy to host this week's RNA collaborative seminar at the Institute of Molecular Biology in Mainz in Germany. I'm Stephanie Ebersberger. I'm a computational postdoc in the group of Julian König, who is the first speaker of today's seminar. And before I will introduce the first speaker, I would just give you some brief information about the seminar. I would like to remind you that this seminar is recorded and that if you have any questions, feel free to post the questions in the Q&A channel, and we will have time to answer some of these questions. And for all the other questions, the speaker will take care to answer after the seminar. And now it is my great pleasure to introduce the first speaker. So this is Julian König. Julian König, he did his PhD in Marburg in Germany, and afterwards he did his postdoc in the group of Jane Uhle at the MRC Laboratory of Molecular Biology in Cambridge, UK. And in the course of his postdoc, he developed iClip, a technology I think most of all are pretty familiar with, and it's a technique that provides genome-wide information of RNA-protein interaction with single nucleotide resolution. And since 2013, he's now a group leader at the Institute of Molecular Biology in Mainz in Germany. And there he continues to decipher how the information stored in the RNA is read and interpreted by RNA-binding proteins. And now I will leave the stage to speaker one and wish you all great fun.
Yeah, hello everyone. Thanks a lot for the nice introduction, Steffi. So today I would like to now it works. So today I would like to talk to you about a joint project between our group and the Sattler group in Munich and the Look group here at the IMB in Mainz. And what we are very much interested in our research is the regulation of splicing, which is the removal of introns and joining of exons to form the mature RNA. And this process is of particular interest because it allows for alternative splicing, which means that certain exons can be either included into the mature transcript or skipped from the transcript, which results in the production of different transcripts and eventually also protein isoforms. And this is a process that is very important, for example, for generating tissue identity, but it's also often targeted in genetic disorders and in particular in cancer. And this is why we want to contribute to the understanding of this process. So two major questions that we have been addressing in the past years is to try to understand more how splice sites can be recognized in this huge sequence space of the pre-mRNA. So pre-mRNAs are thousands and tens or hundreds of thousands of nucleotides long, and splicing has to occur at very defined positions. And also more recently, we became also interested in how the different splice sites can find each other that are also often tens of thousands of nucleotides apart in the pre-mRNA. And so I would briefly like to introduce you the main players that we are investigating. So at the beginning of the intron, the 5' splice site, the U1 SNRP, a part of the spliceosome, is recognizing the splice site in the initial steps of splicing. And at the end of introns, the 3' splice site, two proteins play an essential role. SF1 recognizing the branch point that is important for the catalysis, and U2F2 recognizing the polypyrimidine tract that is immediately upstream of the splice site. And these proteins then recruit the U2 subunit of the spliceosome, which then together with the U1 SNRP initiates further the splicing steps. And so, as Steffi mentioned, in our group, we are doing a lot of genome-wide mappings of protein-RNA interactions to study this process. And one of the approaches that we are heavily using for this in the lab is iClip, which allows us to obtain binding sites transcriptome-wide of a protein in vivo. And what we do is we have living cells, we cross-link protein to RNA with UV, and then we purify the protein of interest together with the RNA with an antibody. And then with help of next generation sequencing, we make libraries of these RNAs and sequence them. And analysis of this data provides us with the in vivo binding landscape. So how does a certain splicing factor act inside cells in the context of a lot of other RNA binding proteins? And this is, of course, very powerful information, but sometimes it's also difficult to deconvolute this data because we don't know, is a protein going to a binding site on its own, or is it maybe brought there by another factor? And to investigate this, we developed in our lab an approach that we call in vitro iClip. And what we do here is that instead of analyzing living cells, we are using recombinant proteins and naked RNA that resemble endogenous pre-mRNAs in this project. We mix this together, the protein binds to the RNA, and then again, with the help of iClip-like purification of the protein and library preparation, we can map binding throughout this in vitro RNAs. And this now gives us the intrinsic binding activity of the protein. So how is the protein binding to RNA if no other cellular factors are around? And then by comparison of this in vivo and in vitro iClip, we can learn about how regulation occurs in vivo. And I want to show you one example of such an experiment that we did for U2F2, this factor that recognizes the pyrimidine tract at the splice site. And here on the bottom is in vivo iClip data. And you can see that this protein nicely binds upstream of the splice site in the transcript here. This is a genome browser view, and here's schematic exons and introns. And when we look at the in vitro iClip data, we can see that some of these binding sites are preserved between in vivo and in vitro, like this one here or like this one here, suggesting that here the intrinsic activity of this protein is enough to explain what it's doing in vivo. But then we also see clear differences. For example, here we have a binding site that is strongly bound in vitro, but it's not bound in vivo, suggesting that some factor is clearing U2F2 binding from this location in the cells. And here we have the opposite, where we have a binding site where in vivo it's strongly bound and a functional binding site, but there seems to be no affinity for this region in vitro of the protein alone. And overall, we see from our analysis that we estimate that about half of these sites are intrinsically recognized, and the other half needs some factors where U2F2 is cleared or stabilized. And we then were interested in what factors could be stabilizing or clearing U2F2 binding in vivo. And to do this, we used a machine learning approach where we took these sites where the binding sites of UTF2 that are stabilized or cleared, and we extracted sequence informations out of windows around these binding sites. These features included, for example, binding motifs of other RNA binding proteins. And then we used machine learning to learn the relevant features that can discriminate between cleared and stabilized binding sites. And this led us then to identify some RNA binding proteins that we suggest can regulate UTF2 binding in cells. And this included, for example, HNRNPC and PTB that were nice positive controls, because we and others had already shown before that these proteins can compete with UTF-65 binding, and consistently we identify these binding motifs here at sites that are cleared in vivo. And then on the other hand, we also identify factors that can stabilize UTF2 binding. And one of these factors was FUBP1. And so, yeah, to summarize what we learned during these initial experiments was, so we have here this core complex of UTF2, SF1, and so on, sitting here at the 3' splice site, but binding of this complex seemed to be heavily modulated by other RNA binding proteins. And in particular, we identify this FUBP1. In parallel, we were also analyzing intrinsic features of UTF2 that help to recognize splice sites. And here we found, using a combination of in vitro clip and NMR experiments in Michael Sattler's groups and other biophysical measurements, that an intrinsically disordered linker between the two RRM domains here contributes to proofreading of pi tracts. But for today's talk, I would like to focus more on FUBP1 and its role in splice site recognition. So, as I said, we identified FUBP1 as a potential new regulator. And so to learn what this protein is doing, we performed clip experiments in vivo with FUBP1. And what we found, to our surprise, in these meta-analyses here, so these are RNA maps or meta profiles of binding around the branch point of all splice sites, of all used splice sites in the transcriptome. And what we found was that FUBP1 actually has a major footprint upstream of the branch point. And this footprint resembles signal strengths that we observe, for example, for UTF2, that is peaking here downstream of the branch point where the polypyrimidine tract is located. And in fact, what we see is that more than 90% of a 3' splice site, that we estimate that more than 90% of 3' splice sites have significant binding of FUBP1, suggesting, and this we sketched here in this graph, that FUBP1 might be a new core component of this 3' splice site recognition. And we wanted to know if FUBP1 also interacts on the protein side with these different 3' splice site factors. And for this, we used BRET, so bioluminescent energy transfer, where we have a donor protein and an acceptor tagged with nanoluciferase and a fluorescent dye. And if these proteins are in close contact, we see this energy transfer that we then can visualize here as this BRET signal. And we did this experiment for FUBP1 and UTF2 and SF1. And indeed, we found that in cells, these two proteins seem to interact, or FUBP1 seems to interact with these other two proteins located at the 3' splice site. And we analyzed this further with a series of biochemical experiments including band shift assays, ITC measurements, and also NMR. And what we found is that these interactions seem to be direct. So on the one hand, we have here these AB boxes at the C terminus of FUBP1 that interact here with these proline-rich regions in the tail of the SF1 protein. And on the other hand, we have here this N-terminal alpha helix, which interacts with the RM2 of U2F2. And I don't have time to show you all this data. I just want to show here exemplary one NMR experiment that we did to show the interaction between the N-terminus and RM2. And what we do here is we have U2F2 protein, and then we titrate in this FUBP N-terminal box. And you can see here nice chemical shifts that are indicating that this FUBP-N box is binding to the U2F2. And when we mutate here a single alanine that is here in the interface between FUBP1 and U2F2, these chemical shifts are lost, suggesting that we lose the specific recognition of this N box via the RM domain. And we are currently in the process of calculating the structure of this interaction based on our NMR data. Okay, so all these experiments came down or boiled down to a model where we have FUBP1 here as an additional factor at the 3' splice site, interacting with SF1, interacting directly with U2F2. And what we also found is that the KH domains bind to UA and UG-rich motifs in RNA that we also find enriched here upstream of the branch point at 3' splice sites. And we wondered, okay, what could be the function of FUBP1 at these splice sites? And we think one aspect is that FUBP1, via these multivalent interactions with several factors, can stabilize the 3' splice site complex. And we tested that again with an in vitro iClip experiment, where we have a library of 2,000 short RNA regions that fit to 3' splice sites. And we incubate them here with U2F2, and then also with U2F2 and FUBP1 in different concentrations. And this is an example screenshot of one of these individual RNAs analyzed, and you can see that U2F2 alone shows very little binding, but when we add FUBP1, we see binding, and if we add more FUBP1, we see stronger binding here. And this binding also in this example fits nice where we observe U2F2 binding in our in vivo iClip experiments. And yeah, this is not an individual example here. So here is the analysis of these 2,000, or actually, I don't know if it's 1,000 or 2,000 anymore, but different splice sites. And you can see here the signal for U2F2 alone, and here the signal for U2F2 in the presence of FUBP1. And you can see that the majority of these oligos are up from the diagonal here, suggesting that they are better bound by U2F2 in the presence of FUBP1. And yeah, so we conclude from this that FUBP1 helps to hold together this complex here. So why could that be important, or are there specific exons where this may be more relevant for? So we analyzed our FUBP1 in vivo iClip further, and what we found was that we see more binding of FUBP1 to splice sites of longer introns. And this you can see here, that for these long introns, we have very strong signal of FUBP1 in this meta profile, and it goes down the shorter the introns are. And this is not only seen in the FUBP1 binding signal, but also here in these FUBP1 motifs. So we have UUA and similar results we have also for this UUG motif. We see that this is not really enriched, this motif, for shorter introns here shown in black upstream of the Blanche point, but for longer introns we see an enrichment of these foot-bound motifs, suggesting that maybe for this long introns it's particularly important that FUBP1 supports the formation of this early spliceosomal complex. Now, we tested whether, or we also wanted to know if it's functionally relevant for the splicing of longer introns, and that's why we generated an FUBP1 knockout cell line with CRISPR, and also an FUBP1 mutant where we have a mutation within this N-terminal alpha helix that interacts with UTF2. And when we then see which exons are less included upon FUBP1 loss, we see that these exons are generally longer than control exons or exons that are more included, suggesting that FUBP1 might be particularly required also functionally for the recognition of exons that are flanked by long introns. We also investigated this a bit from an evolutionary perspective and looked for the presence and absence of FUBP1 in different species. And what we found is that FUB is generally there, suggesting that's an ancient protein, but it seems that it has been lost in several fungal and also protist species. And when we compare now the median intron length in species where FUB is present versus it's absent, we see that if FUB is absent, that these species usually only have rather short introns, fitting to our model that FUBP1 might be required for the splicing of long introns. So, so far I talked mostly about this very early 3' splice site complex, the E complex, where SF1 is present, but during the splicing cycle, SF1 is released and then replaced by the U2 SNRP that binds to the branch point together with the component SF3B1 that we also analyzed in our CLIP data. And what we know from analysis of the FUBP1 CLIP data is that FUBP1 other than SF1 is present still at later stages of splicing and likely at least until the formation of the lariat, so the first catalytic reaction of the splicing complex. And we wondered what FUBP1 could maybe do during these later stages of splicing. And so one possibility would be that FUBP1 also interacts with components of the U2 SNRP or with components of other SNRPs. And what we found by analyzing published data, but also with our own BRED experiments and NMR experiments, is that in addition to these U2 associated and U2 SNRP proteins, we see interaction of FUBP1 also with U1 associated proteins. And this is shown here for these BRED experiments. So it looks like that for SNRPA and SNRP70 and PRP40B, we also see significant signals in these BRED experiments, suggesting that maybe FUBP1 might also be involved in bridging across the intron and communicating with the U1 SNRNP. So we also further dissected these interactions with NMR to check which regions of FUBP1 and these proteins might be relevant. And I don't show the data for this here, but what we see is that likely the AB boxes that are interacting with the proline-rich regions in SF1, that these AB boxes can now also interact with proline-rich regions in the intrinsically disordered regions of these U1-associated proteins. And yeah, one hypothesis is now here that bridging of FUBP1 to these U1 SNRP components contributes to definition of these introns, or to help bridging across these introns. And this might also be something that is particularly relevant if you have longer introns. Also, these longer introns are usually defined by exon definition, and here SF1 is then usually already released before the communication across the intron. And we think that here maybe FUBP1 might also be involved in taking over some of the functions that are normally carried out by SF1. But this goes a little bit in the speculative direction. Okay, and with this, I would like to thank the people that did all the work. So I would like to thank our group, in particular Steffi, who's doing all the bioinformatics for this project, Miriam working on the experiments, and also former alumni Raymond and Andreas did experiments in our group. Then, of course, Clara in Michael Sattler's group did all the biophysical measurements, NMR experiments, and so on that I showed. Hanzo did all the work for this intrinsically disordered region and also helped with the PUB project together with Santi. And then in the look group, Dalmira set up all these bread assays to test the interaction between FUBP1 and several spliceosomal components. And we also work together with Kathy Tarnack's group and the Legevi group on bioinformatics and mathematical modeling of these processes. And yeah, thanks a lot for listening, and I'm looking forward to discussing with you. And yeah, also RNA Collaborative, of course, a lot of thanks for organizing all this. And yeah.
Thank you a lot for this talk, Julia. I think I will just start with a question.
Mm-hmm.
I see now just a question pops up in the Q&A and I'll read it. So Guillermo Parada asks whether you also checked if FUBP1 binding is related to the strength of the polypyrimidine tract or 3' splice sites. So is FUBP binding stronger for stronger polypyrimidine tracts or stronger 3' splice sites, basically, or somehow?
Yeah. So we tested a lot of things there, and I think Steffi maybe knows it better than I do. I mean, the most striking feature that was indicative of FUBP1 binding was the length of intron, and also Steffi investigated the GC architecture of exons, because this is something that also came up from Jill Astlabs and others recently, is that you have different classes of exons correlating with the length of the intron, but that are probably defined by exon or intron definition and so on. And these two features were the strongest that correlated with FUB binding. I think with the splice site strength and the branch point consensus and the pi tract strengths, we didn't really see a lot of correlation. I mean, we expected also, okay, if FUB is there, maybe the pi tract is weaker or something, but I think this we didn't see.
Yes, I think I can confirm this. And there's another question, I will just read it. You have showed that some introns have been spliced more efficient and some less efficient with the while so with the knockout of FEP1, how can you explain this?
Yeah, so we were also thinking a bit about that, but what we see is, in particular for the CRISPR knockout of FOP, is that the introns that are more included usually have shorter introns flanking it. And this means that they would not at all depend maybe on FOP. And then these and then, but the skipping, yeah. So you can, yeah, this is a bit, I should have had a schematic for it. But so the inclusion splice junctions are very short then, and they don't depend on FOP. But the skipping junction is longer, and this depends on FOP. And so there is a, like, the short splicing events are then still happening equally nice in the FOP knockout, but the long one is, and then you get more inclusion. So that's the explanation for more inclusion. And the less inclusion is actually a little bit less intuitive, because you have three. So if you have the splice side, all introns are long, you think, I have long, all depends on FOP, what is the outcome? That's then actually why, so, and this is intuitively not so easy to understand. And this is also, I didn't have the time, but we did some modeling together with the Stefan Legevi group that we are often doing some meta modeling on splicing together. And they found that indeed it's the case if you have 3 splice sites in competition and they are all depending on a factor, then you get less inclusion and more skipping. Yeah.
Okay. Then thank you, Julian. There are several more questions, but I feel we have to move on to the next speaker, otherwise we will run a bit late.
So maybe so maybe you can just put them in the chat and I try to answer them by email. Maybe Steffi, you can give my email, because I need to rush to pick up my son from the Kita. Apologize for not staying for the next talk, but yeah, it was very nice to discuss, and I'm happy to put it to answer it later.
Julian, I will take care of the questions, don't worry. All fine. Thank you very much.
Bye, thanks.
Bye. Okay, then I think Julian stopped sharing, exactly. And it's now my pleasure to introduce the second speaker, and the second speaker is Kathy Tarnack. She also did her PhD in Marburg in Germany, and followed then with a postdoc in a group of Nicolas Lescombe at the European Bioinformatics Institute in Cambridge, UK. And in the course of her postdoc, she developed a computational approach to analyze iClip data. And since 2014, she's group leader at the Buchmann Institute for Molecular Life Sciences in Frankfurt, which is pretty close to Mainz, where she pursues to work at the interface of RNA-protein interaction. And now I will leave the stage to Kathi, and very much looking forward to the talk.
Thank you very much. Okay, so thank you very much for the introduction to present our project here today. This is also a very nice collaborative project that we did together with Julian König and Stefan Legevi at the IMB in Mainz, and also the groups of Andrei Tomastikonenko and Joseph Barash in the US. And it deals with the role of alternative splicing in CAR-19 therapy resistance. So to introduce the disease that we are talking about, this is B-cell acute lymphoblastic leukemia, or short B-ALL, which is a leukemia, a pediatric leukemia, which involves the degeneration and excessive proliferation of B cells. And luckily, there is an emerging immunotherapy which allows to target this disease, and this is the so-called CAR-19 therapy. And the principle here is that T cells of the patients are isolated from the patient and can then be engineered in a way that they express so-called chimeric antigen receptors. So these are T-cell receptors that specifically recognize an antigen of choice. And in the case of the B-all disease, this antigen is the CD19 epitope on the surface of the B cells. And these engineered T cells are then transferred back into the patient where they go and kill the leukemic cells. However, unfortunately, a substantial number of children actually develop resistance to this CAR-19 therapy. And one way to evade the CAR cells is by losing the epitope on the surface of the B cells. And this can occur through alternative splicing of the CD19 gene. And in particular, this involves alternative inclusion of this exon 2 here at the beginning. So if you have inclusion of exon 2, you make a normal full-length CD19 protein, and you have a regular recognition by the CAR cells. However, if the cells change to exon 2 skipping, there's a truncated version of the CD19 epitope, which is no longer exposed on the cell surface and also no longer recognized by the CAR-T cells. And so we were interested to learn more about this splicing event and to understand both the regulation in cis, so to learn about patient mutations that change the alternative splicing, and also learn about the involved splicing regulators. And in the first step, to test whether there are actually patient mutations that change the alternative splicing, we developed a mini gene construct shown here with the alternative exon in the middle and the two flanks in constitutive exon, and then introduced several patient mutations that were presented in this study in Nature Medicine in 2018 that were found in patients after CAR-19 relapse. And then we tested the effects of all of these mutations on alternative splicing in our mini gene. And as you can see here, so this is the splicing of the wild type mini gene, and you have several isoforms that are being made, most of them here at the top, the normal exon 2 inclusion, but then also intron retention and a little bit of exon skipping. And then we find several patient mutations that actually lead to a drastic change in this splicing. So for example, you have a mutation here that leads to very strong intron retention in our CD19 gene, or also you have cryptic isoforms arising here. And so this gave us the idea that there can indeed be patient mutations that change the CD19 alternative splicing and thereby contribute to the CAR-19 therapy resistance. And to systematically map these mutations, we made use of a high throughput screen, which we developed already in 2018, which allows us to measure the impact of mutations on splicing in a high throughput manner. So the idea is that rather than introducing targeted mutations, we take our mini gene and do a random mutagenesis. So we obtain different sets of mutations in this library of different mini genes. And this is then transfected as a pool into human cells where the mini genes will be transcribed and spliced, and you get all the different splicing products. And importantly, we can connect back our splicing products to the original mini genes because they carry a specific barcode sequence. So in reality, it's not just 3, but actually 15 nucleotides that sit at the end of each mini gene and also appear in the splicing product and allow us to map these two, link these two levels of information. So because it's a random mutagenesis, we first perform a targeted DNA seq to actually map the mutations that were introduced in our library. And yeah, we find thousands of mutations across the whole mini gene. They are roughly evenly spread out, as you would hope, with a random mutagenesis. And on average, each position is mutated 28 times in 28 different plasmids, which is good for us to have some reproducibility in the data set and actually check for the variance in our measurements. We also, in addition to the mutated mini genes, we spiked in wild type mini genes, several hundred into the library. So they don't carry mutations, but distinct barcodes. And these wild type mini genes allow us later to estimate the baseline splicing in our assays, and also to learn about the biological variance that we have between the same constructs, but different barcodes. Similarly, we also performed a target DNA seq to measure the splicing outcome, and from our targeted RNA seq to measure the splicing outcome. And from our RNA seq reads, we can reconstruct all the different isoforms that were in our mini gene. So of course, we have inclusion and skipping of the alternative exon. We can also have different versions of intron retention, and we also get cryptic isoforms like you will see in a second. So, and by bringing these informations together, we can now know for each of the mini genes that we had in our library, the splicing outcome that we get in response to these particular mutations. But as you can see here, we have sets of mutations in each mini gene. And so for this, we teamed up with Stefan Legevi, and in his group, they developed a mathematical model which actually allows us to take apart these mini-gene methods and estimate the effect of the individual point mutations on CD19 splicing. And if you look at the outcome of this, you get a profile like here. So in total, we find 350 mutations that show a significant impact on CD19 splicing. And you can also see here the impact on the different isoforms. So for example, here at the top, you can see that we have many mutations around the alternative exon that actually reduce exon 2 inclusion and elevate the skipping of our exon 2. We also find mutations that are involved in intron retention or in alternative splice site activation here at the end. So we have all these mutations, but what we also noticed when looking into our library, that in addition to these 5, what we call sort of major isoforms, we had a lot of cryptic isoforms coming out of our library, because it appears that there are many mutations that actually generate cryptic isoforms in this cryptic splice sites in this region, and they then lead to the generation of these cryptic isoforms. And in total, from our small mini-gene, we find 101 different isoforms that occur. And as you can see here by the color coding and also in this pie chart, most of these isoforms are actually not translatable. So they have either a stop codon or introduce a frame shift. And so most likely all of these cryptic splicing events, if they occur, they will prevent the presentation of the CD19 epitope on the B cell surface. So I would like to use these cryptic isoforms also to tell you a little side story that we came about in our project. So there was one cryptic isoform that we were particularly excited about, and this is the so-called exon 2-part isoform. And this is an isoform that had been described before for CD19, and it's actually a small part out of exon 2 that is spliced out in this isoform. And this isoform had been reported in multiple studies, and it had even been suggested as a potential biomarker. And because it was such a nice non-canonical splicing event, we wanted to learn more about it. These type of sort of excisions from an exon, they are actually called excitrons. So it's like a small intron that occurs inside an exon, and they have been reported quite a lot recently. And so we did a reporter to study the regulation of this non-canonical assay, but somehow nothing ever worked really out. And our results were never really consistent until we realized that this putative excitron here, the small part that is spliced out of our exon, is in fact an artifact from the reverse transcription reaction. And one way how we could show this was by using long read sequencing, so nanopore sequencing, because here you can sequence with Oxford Nanopore either cDNA, so this is after reverse transcription, but you can also directly sequence the RNA. And by comparing Oxford Nanopore data from the same samples, which were either cDNA-seq after reverse transcription or direct RNA-seq, we could find not only in our CD19, but also in several other genes, these artifacts that we see. So you have an intron that is being spliced out of the exon that only appears in the cDNA, but there's not a single splicing event seen in the direct RNA-seq, because this is really an artifact of the reverse transcription. And we also termed these falsitrons, these false excitrons that we find. And by these comparisons of the Oxford Nanopore data, we found a couple of these events that we could directly detect. And what we saw for the CD19 and also these other events, that it appears that there's a strong secondary structure. And then at the base of the secondary structure, you have a repeat sequence. And it seems, or our theory is, that maybe during a reverse transcription, the reverse transcriptase sort of does a template switching here and jumps over this region and just continues at the other repeat. And with this information, we actually went into a publication, data from a publication that just came out at the time, where they mapped thousands of these excitrons in the Cancer Genome Atlas data, and they really found 1,000 of these excitrons. And then we checked whether these excitrons that they detected also had these repeats. And indeed, this is what you can see here in these orange lines. They have the repeats to a larger extent, and the repeats are even longer than on the cases that we had found on the Oxford Nanopore. So we really think that these falsitrons, these RT-PCR artifacts, are something that occurs quite commonly in any type of data set that uses reverse transcription. So it's Illumina sequencing, but it's also long read sequencing if it uses reverse transcription. And they are usually characterized by these repeat sequences, but of course you can think of ways to correct for that, either in your analysis. I mean, here now for the CD19, we corrected bioinformatically afterwards for this isoform, but of course there could also be experimental solutions for that. And if you want to learn more about this artifact, we actually published it in a very nice publication where you can learn more about how it arises and also how we found it. Coming back to our mutations in the CD19, so we now have these 350 mutations that seem to have a strong impact on CD19 splicing. And of course, we wanted to know who are the regulators that are involved in this regulation. And to identify these, Mariella actually built up a very nice bioinformatics pipeline in which she first predicted binding sites for different RNA binding proteins in our CD19 mini gene. Then she looked how these overlapped with the splicing effective mutations, and in the end also checked which of these RNA binding proteins would actually be expressed, both in our patient data and also in the cell line that we use for our experiments. And from this pipeline, we came up with a short list of 11 candidates, which were then tested by Laura in the lab, whether they can actually change the CD19 alternative splicing. Yeah, sorry, we use different tools also for the in silico predictions. And as you can see also here, they often disagree. But still, we came up with this very nice short list. And this is the experiments that we did. And sort of as a positive control here in the front, you can see the knockdown of the SRSF3. This factor was the only RBP that had already been shown before, that it can actually regulate CD19 splicing. And we also recapitulate in our assay that it actually leads to increased skipping of the exon CD19 exon 2 in the SRSF3 knockdown. Another factor that we found that we were also quite excited about was the PTBP1 here, because if you knock down PTBP1, you also get CD19 missplicing, but you actually get a huge increase in intron retention in these sites here. And this was quite nice for us to see, because we see a lot of intron retention in this intron 2 also in the BALL patients. And so we wanted to check whether this intron retention can indeed interfere with CAR-T9 with the surface exposure of the CD19 epitope on the B cells. And so for this, Manu in Andre's group performed a very nice experiment in which he detected CD19 on the surface of the cells and then did a FACS staining and sorting of these cells. And we can actually see that the knockdown of PTB leads to a strong reduction and significant reduction of the surface exposition of CD19 epitope on the B cells, which would indicate that indeed a loss of PTBP1 could lead to reduced CD19 epitope exposure and thereby reduced targeting by the CAR-T19 therapy. So does this also happen in our patients? So of course we checked for the PTB expression in the patients. And for PTBP1, we actually got not really clear results because it went up in some of the patients, down in others, and it was not really clear, but the picture got a lot nicer when we actually looked at the PTBP2, because PTBP2 shows a really clear upregulation in the BALL patients after the CAR-T19 therapy, which could indicate that it could indeed be involved in this therapy resistance or the relapse of these patients after CAR-T19 therapy. And then if we check whether there's a connection between the PTBP2 and our intron 2 retention events in the patients, we actually also saw a strong correlation between PTBP2 expression in the patient and intron 2 retention. And together from this data, we conclude that the upregulation of PTBP1 in these patients under CAR-T19 therapy could indeed be involved in the therapy resistance, because we think that this upregulation could result in increased CD19 intron retention and reduced CD19 epitope presentation on the cells. And with this, I would like to summarize. And I think what we found in this study is that the relapsed BALL patients, so the patients that develop the resistance to the CAR-T19 therapy, that they really show an increased CD19 missplicing. We find many mutations that lead to the production of cryptic isoforms, and in particular also the intron retention in this intron 2. And in general, I think these high throughput screens are really nice to map all these different regulatory elements and also the involved RNA binding proteins. And for our particular event here now, we propose that actually the dysregulation of PTBP1/PTBP2 could be involved in this CAR-T19 therapy resistance. that we see in these BOL patients. And of course, this is what we would also like to follow up in future analysis. And with this, I think that these high throughput screens like we do here, they can be a nice way, like I also showed you, to learn more about a specific splicing event, like here, the CD19 splicing in CAR-T19 therapy. And that's of course something that we want to follow up now in patients. But the same data can also be used for more mechanistic studies. And I did not show this here, but we can also use this type of data to learn more about the binding of RNA binding proteins and the dynamics of regulation that we have in our mini chain by combining the information on all the different mutations that we have. And with this, I would like to thank the people that were involved in this study. So the experiments that I showed you today were mostly performed in Julian König's group, and the bioinformatics analysis were performed by Mariella. Mostly Laura did the experiments together with people from Andre's group and Joseph's group, in particular Manu did the very nice fax analysis for our revision. And then there's Stefan Legewi's group, now in Stuttgart, primarily at IMB, who developed the mathematical model that is behind these data. And with this, I'm finished and I'm happy to take questions.
Thank you, Kathi. Thanks a lot for this great talk. I'm just watching if there are any questions. Oh, here it comes. The first question from Svetlana Lebedeva. Very elegant work. I'm amazed by the RT artifact. I should read the paper, but do you think this might be in ESTs too and result in spurious annotations of introns in 3' UTRs?
I mean, I think it's a good question. I think in principle, yes, they often don't harbor splice sites, but they have these repeats instead. And I guess that this would be a criterion often to not make it into annotation, because there are no splice site consensus sites. There's really instead these repeats on both sides. But in principle, I think they could occur. It was also something that we followed up at some point, but I don't remember exactly what the result there was. But we were looking at that because, of course, you have these 3' UTR introns that are supposed to be regulatory events, and some of them could also come because they are also very short often. Yeah.
Okay, thank you. Then I would also have a question. I wondered when you did your mini-gene mutagenesis, there is, first of all, how many mutations did each mini-gene have on average, and did you assess whether the effect of these individual mutations is additive, multiplicative, if it's always the same?
So on average, they had, I think, 6 mutations in this study, and the previous library 4. We actually went for quite some mutations per mini-gene, because otherwise you have too many mini-genes that A, do not carry any mutations, and B, you just need too many mini-genes. So we rather need sort of the full complexity, and then we take it again afterwards. I think part of it is that it works because many mutations do not have an effect, but the model can also take those apart that do have an effect. I think in a direct correlative way, the exception to that is, of course, for example, mutations in splice sites, because they are no longer additive. So we had certain types of mutations that we had to take out from the model because they sort of break this assumption.
Yeah, I see. So in hotspot regions, so where you would expect that it,
like if you hit a splice site, it's dead, and then it can no longer
I see.
be modulated by any other mutation.
Okay, thank you a lot. So I think we are actually all very happy here right now and perfectly in time. It's 2 to 5, so at least here in Germany. So I would like to thank once again all speakers. And I was very happy that we could host this week's seminar here in Mainz. And yeah, I would like to wish you all a nice remaining day, and see you then at the next seminar in 2 weeks. Thank you
very much.
And goodbye.
Bye.